I'll introduce Ryan Thum. He is, or Thum, he's a professor in plant pathology here, and he does a lot of aquatic weed biology. By the way, this course will be taught in the snow semester. We have tapes of all these lectures. So if you have any friends that want a good, interesting course, they can sign up for basically it's called the spring semester when you sign up for it, but it's a snow semester. Okay, Ryan, go ahead.
Great. Well, thanks, David. And I appreciate the opportunity to be here and talk about my research. Hopefully you guys can see what's on my screen. If there's any issues with that, maybe Duncan or David can chime in at any point along the way. Yeah, as David mentioned, I work on, I guess I would consider myself an aquatic weed biologist on one hand, and then on the other hand, I consider myself an evolutionary geneticist. And so what I'd like to talk to you about today is sort of the direction that I think aquatic weed management is moving. Now that's a very specialized field, but weed management and pest management writ large is a very large field. And so I think everything I'm talking about here today applies more broadly to all of that stuff. And so it's subtitled Personalized Medicine for Lakes because I think many of you have heard of the term personalized medicine. And I think that the direction that this stuff is moving, over the next few decades will be really precise in how things are managed. So, and then as Dr. Sims mentioned, I'm in the plant sciences and plant pathology department, and I'll have my contact information at the end of this if you want to reach out at all. I'll start off with the two, sort of broader main philosophical points that I would hope students would take away from this today. The first is that meaningful evolution can occur on contemporary timescales in our own lifetime. So many of us, I think, have an intuitive perception of evolution occurring on these very long timescales, and that's of course true. But there's lots of rapid evolution in our world as well, in particular when environments change. And then the second thing, I'm partial to this field of population genetics. I teach a course on that here at MSU, and I hope, and I'll be sort of bringing in population genetics in small ways throughout the talk. And so hopefully one thing that you'll be able to take away from the talk is that population genetics is a meaningful tool that's relevant to sort of ongoing and rapid evolution in ways that relate to things like management. I've used this term contemporary evolution, so I'll just stop to define it real quickly. It's just simply rapid evolutionary changes that occur on ecological timescales that have relevance to us in our own lifetimes, in contemporary time. So I'll give you a few examples of that outside of my research that I think you'll be able to identify with. The first is that artificial selection and genetic engineering for crop improvement goes on all the time with plants and animals, to the point today where we can actually rewrite the genetic code for particular genes. This is an example of maybe breeding in some disease resistance in potatoes, let's say. And so my dog, I apologize, I'm doing this from my living room. Aggie, enough. Hopefully we'll get through this. So that's an example of rapid evolution, and that's human-directed evolution. My dog maybe being an example of that. For human health, we've seen rapid increases in antibiotic resistance over time, right? So this is a graph going back to the 1980s to the early 2000s with 3 different human pathogens that are we use antibiotics against widely. And of course, as we use antibiotics, that changes the environment for the pathogen, and then there's rapid adaptation to that environmental change. And so we've seen increase in antibiotic resistance over time. This might be an interesting one, these 2 examples, for those of you who are interested more in organismal biology, maybe fish and wildlife management. This is the Atlantic cod, a very, by all accounts, tasty fish. And historically Atlantic cod matured reproductively. This is a sort of long-lived fish. They wouldn't really start reproducing until around the age of 7. Of course, during that whole time before they're reproductively mature, they're growing. And as there's been lots of harvesting of Atlantic cod, that has actually imposed a selection pressure for earlier reproduction in Atlantic cod, because if they delay the onset of reproduction until year 7 or so, they're likely to get captured and eaten and killed before then and not leave any offspring. So as there's been selection for this earlier reproduction, sexual maturity and reproduction, well, that necessarily means that the fish are smaller when they sexually mature. And so the age of sexual reproduction, or the age of maturity, has decreased steadily over time for Atlantic cod. And as a result, Atlantic cod are smaller than they were historically, which means that we harvest less biomass than we did previously. So that's just a really interesting example of sort of non-intuitive side effect of evolutionary response by a species to harvesting. Similar example with excuse me, bighorn sheep. This graph is a little bit complicated with the wording, but suffice it to say that for a number of decades, the horn size on rams was decreasing. And then in about 1996, there were some hunting regulations that changed, and we've seen a concomitant increase in horn size as a result of the changes in those harvesting regulations. And so, again, we see rapid, meaningful evolutionary change in all walks of life out there. I'm going to be talking to you about management of pests and weeds, and aquatic weeds in particular. Suffice it to say, I won't spend a lot of time on this graph, but suffice it to say that as we introduce new pesticides, in these cases different herbicides with different modes of action, as we develop and introduce those and start to use those in a widespread manner, we see the number of documented cases of resistance to those herbicides increasing. So this is a very fascinating area of research, at least in my opinion. It's what I'll be talking with you about today. But it's a major concern for agricultural production. There's a heavy reliance on herbicides, and it's pretty clear that if you use it, species will adapt. So moving into my research program, I'll give you a little tour of my research program. The premise of my research program is very simple. It's just simply that genetic variation can influence management outcomes. Again, I'm talking about weeds, the response of weeds to things like herbicides. And so this premise is that we wouldn't expect every individual plant to respond identically to a given herbicide treatment. And that's what we see. We see variation in how individual plants respond to herbicides. And so in as much as that variation is determined by genetic variation, then it stands to reason that we can use that genetic information ultimately to predict management response. And if we can predict management response, then presumably we can be more strategic about how we do it and improve our outcomes. I use this analogy a lot of times. What we're trying to do over the coming, really, it would be over the course of a few decades, is do things that are similar to 23andMe or the health add-ons, things like 23andMe or Ancestry.com's version of it, right? So you swab your cheek cells, you send it in, they extract DNA from those sloughed off skin cells and look at thousands of molecular markers. And they can use those molecular markers to predict the chances that you have a certain health condition, or whether or not you're a known carrier for certain inherited conditions. And so this is really sort of the basis of personalized medicine, the recognition that every individual human is genetically distinct, and with the exception of identical twins. And because they're unique, right, that not all individuals may respond to their environment in the same way. And so if we have a very precise understanding of how individuals' genomes translate into their phenotypes, in particular with respect to their health, then we can, in a very precise way, do treatments for individuals. So by analogy, this is what we're looking to do with weeds. I'll give you a really clear example of this. This is Floridone is an herbicide that's commonly used in aquatic plant management. This plant here is Hydrilla. It's an aquatic plant. It's out of the water here, but it's a very commonly managed aquatic invasive weed. Hydrilla can be distinguished, these different strains can be distinguished in how they respond to fluoridone. So this is this graph represents what's called a dose response assay. So you expose plants to increasing levels of this herbicide fluoridone, and you measure their growth. Don't worry about the words so much here on this axis. You can think of this as just growth. And so the normal wild type strain is sensitive to fluoridone by 12 parts per billion. It's killed. And these three other strains exhibit increasing levels of resistance. That is to say, you need higher and higher concentrations of fluoridone to kill these particular strains. And through a lot of forward genetic research, we know now that the specific mutations in the DNA that determine how resistant or sensitive hydrilla is to fluoridone. So this is one section of DNA sequence and the corresponding amino acid sequence for these 4 different strains. In one very small piece of a gene called phytoene desaturase, it produces an enzyme that is part of the pathway that fluoridone disrupts. And basically, amino acid substitutions at one amino acid position lead to these different levels of fluoridone resistance. And so because we know exactly the DNA mutations that lead to these different levels of resistance, we can sequence that very small piece of DNA for a number of plants in any lake and really tailor whether or not to use fluoridone, and if so, at what concentration to use fluoridone in that particular lake. So that's a really neat example, and what we'd like to be able to do ultimately is know those causal DNA variants or mutations for all species, for all herbicides. And so that's a really good example, but the reality is that for most species and for most herbicides, we don't know the genes and alleles or mutations responsible for herbicide resistance. And this is why I find it such a fascinating area to explore as an evolutionary geneticist. So there's no single gene for herbicide resistance. Different herbicides have different molecular targets. So the suite of genes potentially influencing resistance versus susceptibility differs for different herbicides. And there's different mechanisms for herbicide resistance. There can be, resistance can come from mutations in target enzymes or target pathways, but it can also be non-target things like the plant could adapt to detoxify the herbicide, or to uptake it or translocate it or move it across the plant at a lower rate. Those would be different mechanisms for resistance. There may be things that are related to just having nothing to do specifically with the herbicide, but simply allowing the plant to deal with stress better, and therefore being able to deal with herbicides. So there's a variety of different potential mechanisms, all of which would likely have different DNA mutations. And then finally, in the case of fluoridone resistance in hydrilla, that's just one gene that controls that. But of course, it can be controlled by many different genes. And so resistance may be monogenic or a single gene or polygenic. It may be influenced by many genes. So this is why there's a research program on this, because it's a lot more complicated and interesting than, and because of that, we don't have all of the answers. So what I'll spend the rest of the talk talking with you about is, one of the practical questions that I have in building this research is, which genes do we monitor? And so, of course, we don't want to wait to do any genetic monitoring until we know everything. We have to kind of chip away at things. And so I'll talk to you about research that we're doing in the short term, which is basically focusing on identifying strains that we can focus on. And then in the long term, we're talking about identifying specific genes. And so I'll walk you through a bit about the sort of technology and the approach that we use to do that. In looking at strains, I'll give you an example from our research where we've distinguished these resistant versus sensitive strains. This is a different species than hydrilla. This is for Eurasian water milfoil, a species that I work a lot on. And we've been doing just very basic population genetic surveys. It's kind of boring in some ways. It's just basic surveys. You go to a lake, you collect some plants, and you do some DNA fingerprinting. But there's one thing that we've discovered in doing that's really important. It's subtle, but it's really important. And that is that we can find the same strains in different lakes. And so these are two maps of Michigan here. You might recognize Michigan. And the different colored pushpins here from Google Earth are just different strains of water milfoil that we can distinguish with DNA fingerprinting. And so we have collections of the lakes that share the same strains, like all of these that share this purple strain here. That's important because if we find the same strain in multiple lakes, then if we know anything about a strain, then we can apply it to the collection of lakes where we find that strain. So I'll give you a couple of nice examples about that. So these are the results these are kind of dose response curves like I showed you for Floridone before, except they're just 2 points now. So each one of these panels is a different strain of water milfoil collected from a different lake. And this is work done by a PhD student of mine, Greg Chorak. This is kind of what his whole PhD dissertation is based off of. And what we can do is we can culture these strains in the laboratory, and then we can plant them into these solo cups here, these keg party cups, I guess, and plant them into these cattle tanks. And then we can treat half of those cattle tanks with Floridone in this case, or leave the other half untreated. And so for each one of these panels, what we have on the y-axis is how much biomass there was of this strain on average across the tanks that it was in. And the left-hand point is the untreated tanks, and the right-hand point are the treated tanks. And so when Floridone sort of works, quote unquote, this value on the right-hand side should be a lot lower than the value on the left-hand side. I lost my cursor there. So this would be an indication of Floridone sort of quote unquote working. We'll look at this first panel. This strain here in purple is the strain that's found in this collection of lakes. And it turns out that it's resistant to Floridone, or at least to the concentration of Floridone that they use in Michigan. And that's because we don't see a strong decrease in biomass, right? Unlike here where we see it. Here, plants are growing just about as well whether they've been exposed to Floridone or not. In other words, it's basic to from the plant's perspective, it's as if they haven't been treated with any herbicide at all. And so we can be confident that when we see this strain in different lakes, and we do see it in different lakes, that Floridone is not the best option for control of that weed in that particular plant. I'll show you the other side of the coin. Here's another strain highlighted in green here. We see this strain in several lakes so far in Michigan, and this one does respond really well to Floridone. So it grows really well in the absence of Floridone and doesn't grow very well at all in the presence of Floridone. So when we see this strain, which we do, not uncommonly, we can say that there's no evidence to suggest that Floridone wouldn't work on that strain. And so that's one way that we're sort of in the short term trying to be more precise about how we manage. We would not have wanted to see Floridone's commonly used, and so there's a good likelihood that it would have been used in at least some, if not all, of these lakes to control Eurasian water milfoil, and we know it wouldn't have worked, right? And so here now we know it will work in these lakes. It won't work in those other ones, and that hopefully, arguably makes things more precise because we have more information to base our management decisions on. So that's what we're doing in the short term, and I can take questions about that at the end of this. As I mentioned with that hydrilla example, what we'd like to do is really discover all of the genes that are important for all the different ways that different species can respond to different herbicides. And so I'll spend the rest of my time walking you through doing that. This is ongoing research in the lab, so I'll refer to my research in a conceptual way initially, and then I'm going to show you an example from some work that's not done from my lab, but that's very similar to what it is that we're doing. But the advantage of that example is that it's completed, and so I can actually walk you through to the end of that. But the basic premise of this is to create what we call genetic mapping populations. So this is sort of a population genetics approach to discovering genes. What you do is you cross a sensitive strain with a resistant strain, because that creates a population that we know, even though we don't know the genes, we know that there has to be copies of genes now in that population. Some of those copies are resistance alleles, and some of those copies are sensitive alleles, right? Because we've crossed a resistant strain, which must have resistance genes, with a sensitive strain, which doesn't have resistance genes. And if you make a second generation from those, I won't get into the details of why, but maybe if you harken back to your thinking about Mendel and his peas and how he saw phenotypes come back in the second generation, that's the reason you make that second generation. And then you have basically brothers and sisters, plant brothers and sisters that have the same genetic background, but differ in this key way. Some of them being resistant to the herbicide, some of them being sensitive. Just like for you, in your families, you may have siblings that differ in eye color or hair color, even though you have the same biological parents if they're full siblings. Then if we do whole genome sequencing on all of those individuals, which we can do, and expose them to herbicides, we can compare the genes that the survivors have versus the genes that those that die have, and through that we can find genes. So I'll walk you through an example of that. Right here, just suffice it to say that what we have in the lab right now is we've crossed resistant individuals with sensitive individuals. We've done that with a couple of pairs of resistant and sensitive individuals. Because female and male flowers are separated on these plants, we can cut male flowers off and pollinate or fertilize female flowers with males of our choosing. We can make a lot of seeds. Here's an example of a seedling germinating or seed germinating, and here are a couple little seedlings in the laboratory. So if we do that with hundreds of different plants, as I mentioned before, we can plant them into these cattle tanks. We can genotype all of them or do whole genome sequencing from all of them and look at the distribution of genes or alleles in all of our plants. This is just a cartoon illustration of that versus only those plants that are surviving. And so by looking at only the ones that survive, we know which ones survived and which ones died, and we can genetically compare those two. I think I'll move on in the interest of time, but the rest of this slide just kind of gives a conceptual illustration of how to do that. But I'm going to move on to an example with insecticide resistance. So this is insecticide resistance in mosquitoes. Mosquitoes are heavily controlled with insecticides around the world because they're vectors for human diseases. And so this is work done by a series of authors. This is not from my laboratory, just full disclosure. It's a really excellent paper. It's really neat. And so if you're interested in it, I can get you the full citation for it if you'd like to look at it. But I'm going to walk you through how this works. So what these authors did is they collected a bunch of mosquitoes from Guyana in Africa, and they know that that particular population that they collected from is resistant to several insecticides that are commonly used. They bring them into the laboratory and they expose them to like 80% strength insecticide as a way to get rid of any of the individuals that maybe aren't fully resistant to those insecticides. And then what they have is a selected line of insecticide resistant mosquitoes, which they're calling the F0, or basically the sort of the null generation. And then they cross these female mosquitoes with males that come from a population where they've never seen insecticide resistance before. Okay, so they're crossing resistant individuals with susceptible individuals to make a population that has both resistance alleles and susceptible alleles. They then cross those again to get a second generation, and then they split all of those individuals into 3 different treatments. A line that they don't expose to any insecticides, and then a line that they expose to quarter strength insecticide, that's what LD25 means, and 3/4 strength insecticide. And so here, what we would expect is all of the individuals will survive regardless of whether they carry resistance alleles or not. And then in these 2 treatments, those resistance alleles should be more important for survival than they are to this unexposed treatment. And that's the population genetics key in identifying the genes, which I'll get to in just a minute. So we start with a high initial frequency of alleles. We increase the frequency of those resistance alleles, cross them to individuals that don't have any resistance alleles. That means the allele frequency of resistance alleles is going to go down. And then by subjecting them to different strengths of treatment, we should see an increase in resistance alleles across these 3 different treatments. Okay. So this is just sort of the laboratory data on mosquito survivorship to just illustrate the point that that they have a population that varies for insecticide resistance. So here we have percent mortality. So they take maybe 100 mosquitoes in a vial and they expose them to an insecticide. For their susceptible population, nearly all of those mosquitoes die, indicating that they don't have any resistance alleles, even though we don't know which genes those resistance alleles come from. We just know they don't have any because they're all dying. Here, this is the resistant population, and so we know that they have resistance alleles at some unknown genes because none of them are dying. And then this is that F2 generation exposed to 3 different insecticides. And what you see here, let's just focus on this one. What you see here is that there's an intermediate amount of mosquito mortality, meaning that some individuals are resistant to the insecticide and some individuals are susceptible to the insecticide. And that's the real key in finding the gene. So this is the sort of money figure, if you will, for identifying the parts of the chromosomes, or in other words, sort of the gene regions that are involved in insecticide resistance. Over here on the right-hand side are the 3 different chromosomes. So this species of mosquito has 3 pairs of chromosomes. And then there's a variety of genes. This isn't all of the genes, but this is a subset of genes that the authors were interested in or using as an example here. And this is just where they're located on the chromosomes. Over here is the population genetic data. This is for each population, we've got our resistant mosquitoes that they bred with the susceptible mosquitoes, their allele frequency at each gene, and then the allele frequency of the F2 that isn't exposed to insecticide or is exposed at quarter strength or is exposed at 3/4 strength. So what should work is that for a gene that isn't involved in resistance, there should be no difference in the allele frequencies that we see between these 5 different groups, because there's no such thing as a resistance or sensitive allele at that gene. Whereas for a gene that is important for insecticide resistance, we should see increasing allele frequency as we move from the untreated mosquitoes to the treated with 1/4 strength to treated with 3/4 strength. And the 3/4 strength mosquitoes should look like, with their allele frequency, should look very similar to the resistant population, whereas the untreated mosquitoes should look different from that initial population. So these 5 or 6 asterisks identify genes for which they saw that population genetic signature that they would expect at a gene that's important for resistance. So what you see is that in each case, you see the shading getting darker as we move from untreated to treated, from the untreated to the more highly intense treated populations. So as they identify these gene regions, they can look in the full genome sequence to see which genes are there and develop hypotheses about which genes might be important in insecticide resistance and by which mechanisms they work. So this is work that we're doing in the lab. I don't have full results. I've just sort of shown some cartoons here. We do have a full genome sequence for Eurasian water milfoil, the species that we're primarily working on, which is a really nice resource for us. And so we can identify every gene in the genome, and we can walk through that process that I showed you with the mosquito. We're doing that right now. This is actually a study that we completed a few months ago and identified these different gene regions that look like they may be involved in resistance to this herbicide 2,4-D. And we're now going through the process of identifying what genes are in these gene regions. This is just sort of a schematic of the entire Eurasian water milfoil genome from nucleotide 1 all the way out to nucleotide 6 million or so. And so what we're doing now is trying to sort of, through bioinformatics, trying to figure out what these gene regions, which genes they contain, and what might be going on, as well as doing some experiments to confirm that those gene regions are actually involved in 2,4-D resistance. And so we're doing this with different herbicides for Eurasian water. I'm going to stop there. This is my contact information. I'll just say a few things to end here with my contact information. I do teach several classes on campus. I teach the general genetics class every other fall. I teach evolution every spring, and I teach the population genetics course every other fall. And so if any of this was interesting to you, I'd encourage you to look at those classes as options as you move forward in your education here. If you find any of that stuff interesting and would like to talk about it, either about my research specifically or the field of population genetics more generally, I'd be happy to talk with you. I do often have undergraduate research assistantship positions in my laboratory. I have a few for this summer, provided that things go in any sort of more normal way, like they didn't last year, including one from the National Science Foundation, which is a really neat opportunity for undergraduates. So there's my contact information, and I'd be happy to talk to you if you're interested to do so. And I think there's time now to take questions and have a discussion. Thank you very much.
Hi, Ryan. This is Dave, and we also work on weeds. And what we found is that the herbicides industry has learned basically to attack genes or enzymes or pathways in plants that animals don't have, such as biosynthetic enzymes for amino acids. In fact, imazapyr and glyphosate attack the two main pathways. Now, is this also a strategy for insecticides? Do you know?
Yeah, yes, it is. So I'm not super familiar with the physiology, but in general, yes. In general, pesticides target pathways that are specific to the pest, and not specific or not shared with non-target organisms, in particular humans. That would be the main concern, right? So all of these insecticides go through a registration process through the Environmental Protection Agency, and I think I think it's more often now by design. I think historically it was sort of de facto that those pesticides that made it through that screening process tended to be those that, in hindsight, we discovered attacked pathways that were specific to the target organism. And so, for example, for insects, right, they have hormones. For example, that ecdysosone, for example, which determines the transitions and molting periods, right? And we don't have that pathway as humans, nor do many other organisms. And so things that target those kinds of pathways and disrupt growth and development would do so in a manner that would be very specific to that target organism and not to humans. Now, that being said, I mean, things like insecticides that are used to target mosquitoes commonly also target other insects as well, right? Because they would share those pathways. But that's a long answer. The short answer, David, is yes. They tend to target pathways that are specific to them. And in that case of fluoridone, that's attacking the carotenoid biosynthesis pathway, which we don't have as humans because we don't produce beta carotene. We utilize beta carotene, but we don't produce it the way plants do.
Class has any questions? So it seems to me that if you go swimming in this lake, you're going to be drinking various herbicides. Is that true? Could you comment on that?
Yes, that's a good. You're asking me these great questions. I probably should have touched on these in the seminar. Yes, so these are injected directly into the water, and there's a process for that as well. So it has to be posted. When this is going to occur, it has to be posted some number of days in advance. And then the different herbicides have different restrictions associated with them. So the things that we would be concerned about with water usage would be things like swimming, drinking water, and irrigation water. And so many of these herbicides can't be used within a certain distance of a well that's used for either drinking water intake or irrigation water intake. And then some of them, and then they have different what they would call tox profiles and restrictions profiles. So some of them have no restrictions whatsoever. They can be used in the water and there's no drinking restriction, no use restriction at all. That's pretty rare, but there are some herbicides that have no restrictions whatsoever. Most of them have some set of restrictions associated with use. Very few of them have drinking water restrictions. That is kind of weird to think about, but they're used at such low levels that there's a drinking water standard cutoff, and they're almost always applied below that drinking water cutoff. And so anyways, that's true not only for herbicides, but for all kinds of things. There's constant monitoring of E. coli, for example, and a variety of other sort of known chemicals. So if you do use a lake, I know we have a lot of students from Michigan, Wisconsin, Minnesota, where there's 40 to 50,000 lakes between those. It is something to be aware of, to pay attention to what has been used or what's being used, and if there's any current restrictions.
I traveled throughout Russia looking for different pathogens, and I ran into a lot of Eurasian milfoil. Very few of them were infected with pathogens, but I did find some. What do you know about possibly starting to use biocontrol with host-specific pathogens?
Yeah, that's a great, that's sort of the ideal control, right? Would be biological control, in particular, something that's very specific to the target. And so, historically, there is some biocontrol with Eurasian water milfoil and with many weeds, and they use specialist insect herbivores typically that are specialists on that particular species. There's a little bit of work on that. I have a graduate student that's very interested in that and basically doing some kind of bioengineering with that particular insect. It's a hard sell with managers because they apply an herbicide and within hours to days they see the effects, whereas with biological control, this isn't a statement about my feeling about it, but people get a little impatient, and biological control usually takes more time. David, I know one of your interests is in using pathogens, so diseases, to plants, of which there's often very specific host pathogen dynamics. There is a little bit of work on that in aquatic weeds, but not nearly as much as I would like to see it. So I know that's an area of interest for you, David, and so I hope that work goes well and becomes a viable strategy. But I think the idea of using pathogens is a great idea. Again, to influence adoption in the industry and by the stakeholders, it's going to have to be very, very specific and very effective, because if I'm honest with you, it's a sort of a social commentary, but I think it's an uphill battle to displace the herbicide approach in weed management, because it's just sort of the paradigm that it's just the current paradigm of operation. So it's a great field of study, and hopefully it'll bear some proverbial fruit.
Let's see if there are any students here that would be interested in going into this kind of field. Raise your hand if you are. We got one. You have any questions? No.
Well, for that student, if you'd like to reach out, please do. You can do so on your own time and on your own terms. That's fine.
What we have found is that once a field like biocontrol versus weed control gets going, the money behind using synthetic chemicals is very high, and it is very hard to then convince the regulatory officials who are used to regulating chemicals that biologicals are really a different thing. And yet, the herbicide industry right now is in a perfect storm. We have
Yes, it is.
herbicide resistance, which you've studied. We have consumer problems. People no longer want to eat Cheerios. And we have lawsuits, some $2 billion of lawsuits against a company that used to be called Monsanto. In fact, Montana used to be called Monsanto State University because of all the research that we did on those herbicides. So we're seeing a change right now, and we're seeing people that actually read labels, but you can't read a label on a lake. There's no labeling there. So it'll be interesting to see if anybody comes up with a way to say which lake is safe and which one isn't. I don't know what we're going to do. So thank you for your talk, and we'll see if you attracted any students.
Great.
Thank you.
Thanks. Thank you. Take care, guys.